The next presentation in our session this morning is entitled, Can Calcite Dissolving Bacteria Promote Pod Growth? And our presenter is Alan Peper.
Hello, everyone. All right. Thank you for that introduction. I am Alan Peper from the University of Georgia Plant Pathology Department. I am co-advised by Dr. Lee Young and Dr. Timothy Brenneman. And my research is looking into calcite dissolving bacteria and if they can promote pod growth. So first off, peanut is very economically important to the state of Georgia, bringing in 46% of all peanut production comes from the state with 624 million in crop revenue. The second highest, however, with that, it has the second highest cost for fungal herbicide control at 60 million and has one of the highest total percent losses compared to other commodity crops. So the peanut plant is very unique in the fact that it is known as a geocarpy, and a geocarpy is means it flowers above ground, however, it produces its fruit below ground. So this is done by the flower being pollinated, and then the flower becomes the peg, otherwise known as a genophore. And then this moves below ground between 4 and 7 days after fertilization with the ovaries located at the tip of this peg. From there, the pod swells and forms. And what is also very unique about the peanut plant is that it is actively uptaking nutrients through the pod, not just through the roots. So one of those important nutrients is that it is taking up is calcium. So calcium is critical for proper peanut formation, pod formation. Without it leads to calcium starvation and can lead to embryo abortions, pops, termination of pod expansion, reduction in yield, increased disease susceptibility, and just lowers the overall seed quality. So a very early study by Harris in 1949 can show that without available calcium in the pegging zone, no pods form. However, with calcium, you have abundance of pods. And another unique characteristic that was demonstrated in this study is that you can see that the peanut plants themselves are very similar or relatively similar, meaning that it is only the pods that really need this calcium for proper development, not just the plant. So the way farmers alleviate this is by applying gypsum to their field, roughly at ½ a ton per acre. They can also apply limestone at the beginning of the season or every 3 years, I believe. However, with limestone, you have alterations in pH, which is peanut plants are very sensitive to pH changes, and so limestone is not used as often. So this kind of brings us to my hypothesis is how can these calcite dissolving bacteria improve calcium availability in the soil other than applying gypsum or limestone? So my first objective or aim is to isolate and characterize these bacteria from a peanut field, test their efficacy to increase availability of calcium in soil, and then test the potential impact of these bacteria on overall pod growth. So you've probably been wondering, what are these bacteria that I keep discussing, and how do they work? So first off, microbes are able to induce calcium precipitation and solubilization, and this is through a process called biomineralization. So as you can see in this figure here, this microbe is able to take carbonate, calcium, and convert it to calcium carbonate, otherwise known as calcium. However, the reverse is possible through these calcite dissolving bacteria. They actually take this calcite or calcium carbonate and transform it into calcium, a soluble form of calcium, and then also carbonate. However, the soluble form of calcium is what peanut pods need to uptake and for proper pod formation. So the question here is, how can we utilize these bacteria to increase the overall amount of soluble calcium in a peanut field? And so this leads to my first test, which was isolating these bacteria. So I isolated these calcite dissolving bacteria from the Blackshank farm in Tifton, Georgia. Oh, sorry. And the soil was collected from a peanut-only rotation field within the pegging zone, roughly 5 to 7 centimeters below the peanut plant. And so the way we're able to determine if this bacteria is able to use calcite solubility, we use a selective media. So this is an opaque media. So calcite is insoluble in water, and this creates an opaque plate. So you can see here that this bacteria, these other bacteria do not form this halo. So this halo or clear zone formation is the actual calcite being dissolved into soluble calcium. So within this halo is soluble calcium. And this is the bacteria here growing. And so based off of this measurement of the colony versus the halo formed, we developed, or we used a formula to create a solubility index. And so with that, we have 4 different bacterial strains below. So you can see here A99 has a very high solubility index and produces a high amount of soluble calcium. So with that, I have isolated and collected 64 calcite dissolving bacteria from the soil. 14 of those have been sequenced so far using Sanger sequencing and using the 16S rRNA genes. I've classified them to the genus level, as you can see on this chart here. However, and they also have several morphological differences, being color, structure, and that kind of thing. However, with this, there are multiple repeats or similar or identical strains within the 64 that I've collected. So the next step was to test them on soil or sand and just see if they're able to naturally increase the amount of soluble calcium. So here in the none, this is just plain sand with nothing added to it. We have our 2 strains that I tested here. So again, A99 seems to be a very high performer. As you can see, this is a almost 3-fold numerical increase in the amount of soluble calcium compared to the none. So this gives us some promise and hope that by applying these bacteria to the soil, it will increase the amount of soluble calcium. So this kind of brought us to another way is how can we efficiently test and see what these bacteria are able to do in a very efficient way. So we wanted to develop a new system to study very pod-specific responses to calcium changes in availability. So most of you have already seen this kind of old system, or it's a very useful system where the pod and the root are separated. However, with this, you're only separating the root and pod, and you're not looking at each pod very specifically. So what we wanted to do was develop a growth in tube system where it's very high in efficiency. It has an identical genomic background, meaning that it only needs 1 plant, and then we can test several different treatments on that single plant on many different pods. And we wanted to be able to control the microbial background because we're looking at microbes and bacteria, and we don't want to have any interference in the results of that. And then also we just want it to be easy to alter at treatments. So over here you can see this tube kind of prototype that I worked out, and you can see it is actually able to grow a pod within this 50 ml falcon tube. And so this is kind of what I called the growth in tube system, and I've developed this over some time. So it's just a 50 ml falcon tube. It can be filled with soil or other media. Has a gas exchange system, and so then this just allows for airflow and moisture control. There's a hole at the end, and this is where the peg is actually inserted to the tip of this tube, and then the peanut will grow inside. And then there's just parafilm on the end to seal it. The whole thing is covered in aluminum foil to prevent sunlight. And also this whole unit can be autoclaved to provide sterilization and eliminate other foreign microbes. So over here we have some sort of some preliminary data. So you can see in the open soil, meaning it grew in a pot, and then over here you can see in the tube. So yeah, there's definitely some distortion within the pod, and this is due to the compaction of the media inside the tube. However, in our future experiments, we've alleviated this issue by using vermiculite as a way to reduce this compaction factor, and have had much more success in forming a normal looking pod. Also, if you're wondering about these brown spots on here, that is just soil that I did not wash off completely. And so down here is some statistics. So this again is the open soil grown in a pot, and then over here is the growth and tube. And these, there is no statistical difference between the two, meaning that the tube is not actually having an effect on the actual pod weight. So again, the tube is then applied to each peg. So again, we can have between 10 and 20 of these tubes on one plant. You could have more, it's just a limitation of space. Yeah, and so kind of summarizing everything I've done up to this point, I've isolated some calcite dissolving bacteria from Georgia soils. These bacteria can increase soluble calcium in plate and in sand. I have sequenced 14 of these bacteria so far, and I've developed a new system to hopefully study all of these bacteria. So in the future, I'd like to complete and do more isolations of these bacteria and then test their ability to improve calcium availability. And the end goal of all of this is to create a shelf-stable biofertilizer that can improve peanut health and soil health and reduce the need for farmers to apply gypsum to the field. Another kind of aim that we're looking at is the application of this bacteria indirectly or directly in control of aspergillus. Is a big problem in the peanut industry, and there have been links showing that calcium can have an effect on the severity of aspergillus infection. So again, with the system, we're able to actually test multiple bacteria all on the same plant, and so then this just allows us for a very clean background and enables us to be very efficient with our experimentation. And so with that, I'd like to acknowledge my lab members, my co-advisors, and my committee members, and also our funding sources, the UGA Startup Fund and the Georgia Peanut Commission. Thank you, and with that, I will take any questions.
Thank you, Alan, for that presentation. We have time for questions. First question we have is from Dr. Rebecca Bennett. She says, how do you keep the medium and the tubes moist, or was this even necessary for pod development?
Yes, so moisture is actually very important for the tubes. If they do dry out, the pods don't form. So with that gas exchange, it is actually very good at controlling the moisture within. So before we seal the tubes, we run sterile water through the system, and this hydrates all the soil.
Okay. A second question is, do you know how the relative abundance or the frequency of those calcium dissolving bacteria, are they common in the environment, or is this restricted to the blackshank farm in Tifton? Do you know anything about the ecology or the relative abundance of these in the natural environment or in the agricultural environment?
Right. So we only did our experiment so far in blackshank farm. However, they are relatively abundant, as we've seen throughout all of the farm, not just the peanut only. We just noticed that there is a higher abundance in association with the peanut field. However, I do believe that they are very abundant in just soils naturally.
Okay. That concludes the questions we have. Thank you very much for your presentation.
Thank you.